Welcome to Dr. Kumar's botany classes. Today's topic we will talk about Mendelism. Mendelism is started by Gregor John Mendel, who is termed as father of genetics because based upon his principle, the branch of genetics got started. Mendel was basically a physicist and mathematician. He used to teach science and mathematics at the University of Brunn. But during his leisure period, he was performing hybridization experiment on garden pea plant, Pisum sativum. Because he was a mathematician and physicist, then his colleagues did not take interest in Mendel's activities because it was out of the way. Biology normally a teacher, an individual who is teaching physics and mathematics, he or she is least interested in plants. But Mendel was much interested in plants and he was performing plant breeding experiments that is termed as hybridization experiment in pea plant, garden pea plant, Pisum sativum. He performed two different types of experiments. One is termed as monohybrid cross. Monohybrid cross. What is that? Cross means cross pollination. Cross means cross pollination. That is transferring pollen grain from one flower to stigma of another flower. Transferring pollen grain from one flower to stigma of another flower. But when we are performing a cross, considering only one character at a time, it is termed as monohybrid cross. A cross performed considering one character at a time. Considering one character at a time is monohybrid cross. Monohybrid cross. Similarly, Mendel performed another experiment which is termed as dihybrid cross. Dihybrid cross. It is a cross performed considering two characters at a time. Considering two characters at a time. At a time. So these are the two crosses that Mendel performed, and based upon his findings, he proposed two principles which govern transfer of character from parents to offspring, and that formed the basis for genetics. That formed the basis of branch of genetics in biology. Monohybrid cross, a cross performed considering one character at a time, is monohybrid cross, while a cross performed considering two characters at a time is dihybrid cross. We will go through these two crosses one by one. On the basis of monohybrid cross, we have got two laws of Mendel, and on the basis of the dihybrid cross, we have got one law of Mendel. Presently, we study that there are three laws of Mendel, and out of those three, first two is based upon monohybrid cross, and the other one is based upon dihybrid cross. So, one by one, we will go through these two types of crosses. Mendel considered 7 pairs of contrasting character for his experimentation. Mendel considered 7 pairs of contrasting character for his experimentation. Mendel considered 7 pairs of contrasting character for his experimentation. experimentation. Number one is length of plant. Length of plant. Number two is flower color. Flower color. Number three is seed color. Number four is pod color. Pod is the fruit of pea plant. Number five is seed shape. Seed shape. Number six is pod shape. And number seven is position of flower. Position of flower. So Mendel studied these seven characters, and these characters in pea plant are found in two contrasting forms. As far as length is concerned, they are tall and dwarf. Tall and dwarf. Flower color, it is purple and white. Purple and white. Seed color, it is yellow and green. Yellow and green. Pod color, it is green and yellow. Green and yellow. Seed shape, it is round and wrinkle. Round and wrinkle. Pod shape, it is inflated and constricted. Inflated and constricted. Position of flower, it is axial and terminal. Axial and terminal. These are the seven pairs of contrasting character that Mendel studied, and he obtained first pure breeding varieties. Pure breeding tall plant, pure breeding dwarf plant, similarly pure breeding purple and pure breeding white. Pure breeding means what? Upon selfing, they will produce the same variety. There will be no change among the progeny. The phenotype will remain as it is. That is termed as pure breeding varieties. So Mendel studied these seven pairs of characters, and he first collected 14 types of pure breeding varieties. 7 for these traits, 7 for these traits. 14 types of pure breeding varieties that Mendel studied. The most common, the first activity, the first monohybrid cross that Mendel performed was regarding the length of plant. We are going for these, the first experiment performed by Mendel, considering the length of the plant body. Length of plant, that is tall and dwarf. Tall and dwarf. These plants are diploid variety. Diploid means a gene is represented by two different forms, two units. A gene is represented by two units. Diploid means what? The chromosomes are present in homologous pair, and those homologous pair chromosomes will contain the same gene at the same locus. Right? So for tall, we take two genes represented as TT, and for dwarf, we take two genes represented as small t. These are the parental. These are the parents. This is the genotype of parent. Genotype of parent. These, the two parents are going to produce gametes of only one type. Gametes of only one type. It will produce gametes of having a small t, and it will produce gamete having only capital T. When an organism is producing one type of gamete for a particular character, that individual is termed as homozygous. That individual is homozygous. It means these tall and dwarf plants, they are homozygous variety. Homozygous means pure breeding variety. Homozygous means pure breeding variety. These two gametes will fuse together, resulting into this combination, that is the F1 generation. It is termed as first filial generation. First generation of this cross. First generation of this cross is this one. And the phenotype means externally visible character. Phenotypes means externally visible character. The phenotype of this individual F1 generation is tall. It does not mean that it is not having the character of dwarfness. You see that dwarfness character is present, but the plant is tall. It means that this gene, capital T, is dominant, is masking the effect of this gene recessive. Recessive gene is completely masked by the dominant gene. That is why F1 generation is having this type of phenotype. When we cross these tall plants with similar variety, this is termed as selfing. This is termed as selfing. When two individuals having the same genotype are crossed, the term is selfing. Now these plants will produce gametes of two different types. One is having capital T, another having a small t. Similarly, this plant will be having one gamete having capital T, another having a small t. These two gametes will fuse with these two randomly. Any gamete is going to fuse anyone. This is termed as random fusion, random matting. If this gamete fuses with this one, the plant is having the genotype this one. If this gamete fuses with this one, the plant is having the genotype this one. Similarly, if this gamete is fusing with this one, the plant will have genotype capital T, small t. If this gamete is fusing with this one, the plant will have genotype small t, small t. This is the F2 generation. This is F2 generation. Second filial generation. These are the gametes of F1 generation. And this is the progeny of F2 generation. This is tall. This one, these two are also tall, as you find in F1 generation. But this one is dwarf. This one is dwarf. So tall is to dwarf. The ratio of tall is to dwarf. Age 3:1. That is termed as monohybrid ratio. That is termed as monohybrid ratio. Tall, tall is to dwarf. Age 3:1. This is monohybrid ratio. Monohybrid ratio. Because Mendel was basically a mathematician, he always kept statistical records of all his experiments. He always kept statistical records of all his experiments. And that is why always he was talking about the ratio. Always he was talking about the number. The number of individuals which are tall in F2 generation are 3, and compared to that, number of individuals which are appearing as dwarf in F2 generation is 1. So the ratio is 3:1. That is termed as monohybrid ratio. So it was the first experiment performed by Mendel. It was the first experiment performed by Mendel. And based upon this experiment, there are two laws of Mendel. First law is termed as law of dominance. Second law is termed as law of segregation. You will find that in F1 generation, both the alleles of genes are present together. But the plant is tall. It means that gene for tallness is masking the effect of gene for dwarfness. That is why the F1 generation, all the members are tall. Then the gene for tallness is dominant, while the gene for dwarfness is recessive. Tallness is dominant character, dwarfness is recessive character. That is the first law of Mendel. termed as law of dominance. Second law of Mendel is law of segregation. Law of segregation means what? In F1 generation, both the alleles are present together, but those two alleles get separated from each other at the time of gamete formation, and these gametes fuse randomly with the gametes of other plants. So these two alleles at the time of gamete formation get separated from each other. Get separated from each other. To get separated is segregation. These gametes get separated from each other. In this progeny, we are finding that both the tallness and dwarfness genes are present. These tallness and dwarfness genes are getting segregated at the time of gamete formation, and they are fusing to form this result, which is termed as monohybrid ratio, that is 3:1. Now talk about dihybrid ratio. When two characters are considered for a cross, it is termed as dihybrid cross. When two characters are considered at a time for a cross, it is termed as dihybrid cross. So talk about dihybrid cross. The first dihybrid cross that Mendel performed was between round yellow seeded variety and wrinkled green seeded variety. Wrinkled green seeded variety. Round yellow seeded variety and wrinkle green seeded variety. Wrinkle green seeded variety. Right? For round, we take capital letter R. For yellow, we take capital letter Y. Because round is dominant over wrinkle and yellow is dominant over green. Round, the seed shape, is dominant over wrinkle seed shape. Round seeded color, yellow seeded color is dominant over green seeded color. Similarly, the gene for wrinkle is a small r, and for green, a small y. At the time of gamete formation, there will be only one type of gamete, that is capital R, capital Y. Similarly, in this case, it is only one type of gamete, a small r, a small y, because the plants are homozygous. Homozygous means pure breeding, producing only one type of gamete. These two gametes will fuse together, forming the zygote, capital R, capital Y, small y. This is the F1 generation. The plants of F1 generations are, because R capital is dominant, it is round, and Y capital is dominant, it is yellow. So all the progeny in F1 generations are round yellow, because capital R is dominant over a small r, round is dominant over wrinkle, and yellow is dominant over green. That is why this is the condition round yellow. If round yellow variety is selfed, capital R, capital Y, small y, now the gametes will be of different nature. There will be four types of gametes produced by F1 plants. In case of a dihybrid cross, four different types of gametes will be produced by F1 plant. The gametes will be, just take these two are different, take two. Similarly, yellow and green, these two are different, take two and multiply. There will be four types of gametes produced by these plants. Similarly, this will also produce four types of gametes. Because capital R for round, small r for wrinkle, these are two different units, take two. Capital Y for yellow, small y for green, they are also different, take two and multiply. 2 into 2, 4. Four types of gametes will be produced by these organisms and these organisms. The type of gamete will be calculated like A plus B multiplied by C plus D, capital Y plus small y. When you are going to find how many types of gametes an individual is going to produce, you have to utilize this formula. These two are different, then take R plus R. These two are different. Take y plus y and multiply. You will find if R is multiplied by y, the gametes are capital R, capital Y. If this R is multiplied by a small y, the gametes are capital R, a small y. Similarly, a small r if taken, it will produce a small r capital Y and a small r a small y. These are the four different types of gametes produced by these plants. In this case also, four different gametes, capital R, capital Y, one capital R, a small y, another a small r, capital Y, a small r, a small y. These four gametes will fuse with any of these because there is a random mating there at the time of fertilization. Any of these four gametes are going to fuse with any of these. Because there are four gametes and this plant is also producing four gametes, 4 into 4, there will be 16 zygote. There will be 16 zygote. These 16 zygotes will be explained by formation of a checkerboard which is termed as Punnett square, developed by British botanist R. Punnett. So now we are getting the result of F2 generation with the help of Punnett square. We will get the result of F2 generation with the help of Punnett square. Important thing is that in case of David cross, you must be knowing how to decide the number of gametes produced by an individual. How to decide the number of gametes produced by an individual. Suppose an organism is having this genotype. Suppose an organism is having this genotype. How many types of gamete they are going to produce? This capital A, small a, different, take 2. Capital B, small b, different, take 2. Capital C, small c, different, take 2. Capital D, small d, different, take 2. And multiply. Now it is 2 into 2, 4 into 2, 8 into 2, 16. It will produce 16 types of gamete. Similarly, take another genotype. Suppose it is this one, this genotype. How many types of gamete it is going to produce? These two are different, capital A, small a, take 2. These two are similar, capital B, take 1. These two are similar, capital C, take 1. These two are similar, small d, take 1 and multiply. So individual having this genotype will produce only two types of gametes. It means that don't go for the length of the genotype. How many genes are concerned? We don't consider. We will consider how many genes are of different nature. How many genes are of heterozygous condition. We say the term heterozygous. When the two genes are different, two alleles are different, we are using the term heterozygous. So this dominant allele is this is the recessive allele. When these two are different, it is the heterozygous condition. So this genotype is actually monohybrid. Heterozygous for one character, the other characters are homozygous. This is tetrahybrid. All the four characters are heterozygous. When the characters are heterozygous, when there are two different alleles for the same gene, the condition is termed as heterozygous. Now in this case, all the four genes are heterozygous in nature. That is why we are taking 2 each for each one and the result is 16. But in this condition, in this genotype, you are finding that only this gene is heterozygous. The other 3 are homozygous. That is why for homozygous condition, I am taking 1. For heterozygous condition, I am taking 2 and multiply. Now it is only 2. Only 2 types of gametes will be produced by these individuals. Only 2 types of gametes will be produced by these individuals. And that is the condition. So when we are talking about the number of gametes or types of gametes, we have to consider how many genes are showing, how many genes are present in heterozygous state and how many genes are present in homozygous state. Homozygous means what? The two alleles are similar to each other. Heterozygous means what? The two different alleles are present together. So this individual is heterozygous for all four characters, but this individual is heterozygous for only one character. And that is why it is producing only two types of gametes, and this one is producing 16 different types of gametes. Right? And whenever we are going to decide what will be the nature of the genotype of the gametes, we have to use this formula. To decide the nature of genotype of gamete, we will utilize this formula, A plus B multiplied by C plus D. Okay, in this case, you have to take A plus A multiplied by B plus B multiplied by C plus C multiplied by D plus D. Erase the bracket one by one and you will find the type of gametes. It is not a problem. You will find there are 16 different types of gametes. Right? Now we are going to find F2 generation for a dihybrid cross. It means that these four gametes are going to fuse with all these four. Okay, and that will be represented by only Punnett square. That will be easier in nature to provide the result through Punnett square. Now I am going to try the Punnett square for this dihybrid cross. The Punnett square prepared by the British botanist Mr. Punnett for the first time. We take gametes in this X and Y axis. This is like finding coordinates at a particular point. To find the genotype of the zygote is like finding coordinates at particular points. Right? Take the gametes of these plants in x-axis or y-axis are y, capital R, small y, small r, capital Y, small r, small y. Similarly, the other gametes will be produced in this series: capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. This is now coming. If you are going to find the results of these 16 types, it is what? Take this gamete to this position, take this gamete to this position. What you are getting? You are getting the coordinates at that particular point, like in mathematics. So the condition is capital R, capital R, capital Y, capital Y. Here it is capital R, capital R, capital Y, small y. Always try to write capital letter first, then followed by the recessive letters. In this condition, capital R, small r, capital Y, capital Y, capital R, small r, capital Y, small y. Similarly, at this point, capital R, capital R, capital Y, small y, capital R, small y, capital R, small r, capital Y, small y, capital R, small y, small y. At this point, capital R, small r, capital Y, capital Y, capital R, small r, capital Y, small y, small r, capital Y, small r, capital Y, small y. At this point, capital R, small r, capital Y, small y, capital R, small y, small y, small r, capital Y, small y, small r, small y, small y. This is the Punnett square for a dihybrid cross. This is the Punnett square for a dihybrid cross for the fusion of these four gametes with these four units. Okay. Now check the genotype. This is the genotype of the 16 zygote. Now finding the phenotype of these zygotes, we will go through that. Here it is capital R for round, capital Y for yellow. These gametes are present in all these four units. It means these four are having round yellow. These four are having round yellow. Similarly, capital R, capital Y is present in this gamete and this gamete is present in all these four. It means these four are also round yellow. Similarly, you will find that these two units are also having capital R, capital Y. These are also round yellow. So when you are performing a dihybrid cross, these 9 units 1, 2, 3, 4, 5, 6, 7, 8, 9 are always having both characters of dominant type. Are always having both characters of dominant type. So the progeny in F2 generations are round yellow. How many? 9. Round yellow, they are 9. Now talk about these units. Now talk about these units. 1, 2, and 3. These three are similar to each other. R is dominant in this case and Y is recessive. R dominant for round, Y recessive for green. So round green, they are 3. Now talk about these 3 units. A small r, capital Y. A small r, capital Y. A small r, capital Y. It means what? Wrinkle yellow. Wrinkle yellow. How many are the number? It is 3. Now this last one, it is wrinkle green. Wrinkle green. It is only 1. So dihybrid ratio is 9:3:3:1. So now, just when Mendel found the dihybrid ratio to be 9:3:3:1, his mathematical brain started tickling. If we consider these two characters, round and wrinkle shaped seeds and yellow and green colored seeds, individually, what we are getting? Cross round with wrinkle. What ratio we will get in F2 generation? 3:1, because it is the monohybrid condition. Take another unit, yellow and green. Yellow and green individually. When you talk about crossing an individual plant having yellow flower, yellow seeds and green seeds, what ratio we are getting? We are getting 3:1. So when we are talking about round and wrinkle, the ratio is 3:1. When we are talking about yellow green, the ratio is 3:1. When we talk about round yellow and wrinkle green, the ratio will be product of these two units, that is 9:3:3:1. This is where Mendel succeeded and other biologists failed, because those biologists did not care about the number much. But Mendel at each and every step was caring for the number, caring for caring for statistical records. Then what is that? What we are getting? Monohybrid ratio to the power 2 is dihybrid ratio. Monohybrid ratio to the power 2 is dihybrid ratio. It is the basic point which will help you to solve the numerical problems related with Mendelism. Thank you. Thank you for being with me. Thanks again. Thank you.